Dear students, today we are going to discuss an important topic, that is the analysis of sequence data. Let's have a look on today's objectives. First, to acquaint the students about the need of sequence analysis. Second, to tell them about the sequence data management and analysis. Third, to give them a brief idea about the type and nature of sequence submitted and genomic annotation. And fourth, to have a brief discussion about how to perform sequence analysis using diverse software programs and web-based applications. Let's begin with the introduction. Bioinformatics is the application of computational tools to predict, manage, and interpret biological data. It's one of the essential tools for integrative and multidisciplinary understanding of metabolic network processes in systems biology. For instance, understanding omics data requires both common statistical and computing-based methods due to the multidimensional and complexity of the data. Information processing by bioinformatic tools and computational biology methods has become essential for solving complex biological problems in the field of genomics, transcriptomics, proteomics, and metabolomics. Such methods give new insights into areas such as genome evolution, systems biology, biotechnology, genome deciphering, and developments in medicine. Generally, there are three central biological principles around which biological tools must be developed. First, DNA sequence determines protein sequence. Second, protein sequence determines protein structure. And third, protein structure determines protein function. In bioinformatics, sequence analysis is the process of subjecting a nucleotide or amino acid sequence to a wide range of analytical methods to understand its features, function, structure, or evolution. Methodologies used include sequence alignment, searches against biological databases, and others. As a result, there has been a rapid progress in algorithm development in computational biology. Large number of softwares are available to compare an unknown nucleotide or amino acid sequence to all the sequences in the database. Since the development of methods of high throughput production of nucleotide and protein sequences, the rate of addition of new sequences to the databases increased exponentially. However, comparing these newly identified sequences to those with known functions is a key way of understanding the biology of an organism. Therefore, sequence analysis can be used to assign function to genes and proteins by the study of similarities between the sequences under study. Nowadays, there are numerous tools and techniques available for sequence comparisons, that is the sequence alignment, besides to analyze the alignment product in order to understand its biological outcome. Dear students, let's now talk about the data management and analysis. As biological databases are growing in terms of numbers, size, and complexity, managing these databases is becoming very complex. Most of these databases emerged as the output of experimental research by scientists. However, many of the scientists were not trained on the information side of research, and also there were few standards available to be followed in defining these databases. This resulted in the haphazard growth of biological databases, and a lack of integration continues to be a major bottleneck. DNA sequence records from the public databases, that is the DNA Data Bank of Japan, European Molecular Biology Laboratory, and GenBank, are essential components of computational analysis in molecular biology. Integration of data from various sources can be a valuable instrument in understanding and research. For instance, the three-dimensional structure of a protein is more useful if combined with the data about the function, occurrence in different genomes, and interactions with other molecules. If this information is conveniently presented, individual pieces of information are put in context with respect to other data. As an example of a unified database access, consider the sequence retrieval system, SRS. Sequence retrieval system allows any flat file databases to be indexed to each other, and hence it is a system for integrating heterogeneous databases. It's based on pre-made indexes of the items, that is the words, entries, data fields, text and the like, found in a set of documents, that is the database files. Entries. Entries facility provides a gateway to DNA and protein sequences, genome mapping, data, three-dimensional macromolecular structures, the PubMed bibliographic database, and the like. A search for a particular gene in either database will allow smooth transition to the genome it comes from, the protein sequence it encodes, its structure, bibliographic reference, and equivalent entries for all revealed genes. Similar to the SRS system, it provides search facilities for a large number of databases and provides links between them. It provides a well-defined web interface that allows programs or a website to define links that will query entries. However, the entry system is not available to set up at one's own server. It's purely a system for accessing and searching the database at NCBI. Once you have found a way to access the data, you need to understand what needs to be done with this data. It's now fairly efficient and convenient to get the data access, but it is more difficult to define and visualize the kind of operations that are required to convert this data into biologically meaningful analysis. Dear students, let's now talk about the why, where, and what to submit. Presently, it's assumed that all submissions of sequences are done electronically, via the World Wide Web, by electronic mail, or at the very least on a computer disk sent via regular postal mail. The uniform resource locators and email addresses for electronic submissions will be discussed as we proceed in this lecture. The submission process is quite simple, but care must be taken to provide information that's accurate, free of errors and vector or mitochondrial contamination, and as biologically sound as possible to ensure maximal usability by the scientific community. A few things should be considered before starting a submission, regardless of its form. First, the nature of the sequence. Instead of genomic or mRNA origin, users of the databases like to know the nature of the physical DNA, that is the origin of the molecule being sequenced. For example, although cDNA sequencing is performed on DNA and not RNA, the type of the molecule present in the cell is mRNA. The same is true for the genomic sequencing of rRNA genes, in which the sequenced molecule is almost always genomic DNA. Copying the rRNA into DNA like direct sequencing of rRNA, although possible, is rarely done. It should be kept in mind that because the sequence being submitted should be of a unique molecular type, it must not represent, for example, a mixture of genomic and mRNA molecule types that cannot actually be isolated from a living cell. Dear students, we also need to find out that is the sequence synthetic but not artificial. There is a special division in the nucleotide databases for submitting synthetic molecules or sequences put together experimentally that do not occur naturally in the environment. For instance, protein expression vector sequences. The DNA sequence database do not accept computer-generated sequences such as consensus sequences, and all sequences in the databases are experimentally derived from the actual sequencing of the molecule under study. Dear students, we also need to look how accurate is the sequence. The question is poorly documented in the database literature, but it is to be assumed that the submitted sequence is as accurate as possible usually means at least 2 pass coverage in opposite orientations on the whole submitted sequence. Equally important is the verification of the final submitted sequence. It should be free of vector contamination. This can be verified with a blast and search against the VecScreen database and possibly checked with known restriction maps to eliminate the possibility of sequence rearrangement and to confirm correct sequence assembly. Dear students, let's now see the type of organisms that are involved in the sequencing. All DNA sequence records must show the organism from which the sequence was derived. Many inferences are made from the phylogenetic position of the records present in the database. If these are wrongly placed, an incorrect genetic code may be used for translation, with the possible consequence of an incorrectly translated or prematurely truncated protein product sequence. Just knowing the genus and species is usually enough to permit the database staff to identify the organism and its lineage. NCBI offers an important taxonomy service, and the staff taxonomists maintain the taxonomy that's used by all the nucleotide databases and by SwissProt, a curated protein database. Dear students, let's now talk about the coding sequences. A submission of nucleotide also means the inclusion of the protein sequences it encodes. This is important for 2 reasons. First, protein databases, for instance SwissProt and PIR, are almost entirely populated by protein sequences present in the DNA sequence database records. Second, the inclusion of the protein sequence serves as an important, if not essential, validation step in the submission process. The coding sequence features or CDS are the links between the DNA or RNA and the protein sequences, and their correct positioning is central in the validation, as is the correct genetic code. The nucleotide databases now use 17 different genetic codes that are maintained by the taxonomy and molecular biology staff at NCBI. Because protein sequences are so important, comprising one of the main pieces of molecular biology information on which biologists can compute, they receive much deserved attention from the staff at the various databases. A submitted record will be validated by the database staff, but even more immediately by the submission tool used as well. Validation checks that the start and stop codons are included in the coding sequence intervals, that these intervals are using exon-intron consensus boundaries, and that the provided amino acid sequence can be translated from the designated coding sequence intervals using the appropriate genetic code. Dear students, let's now talk about the genome annotation. Genome annotation is a process that consists of adding analysis and biological interpretations to DNA sequence information. This process can be divided into 3 main categories. First, annotation of nucleotides. Second, annotation of proteins. And third, processes. Annotation of nucleotides can be done when there is information about the complete genome or DNA segments of an organism. It involves looking for the physical location, that is the position on the chromosome of each part of the sequence, and discovering the location of the genes, RNAs, repeat elements, and so on. Annotation of proteins involves searching for gene function. Besides, general predictions about gene and protein function, other information can be found in an annotation, such as biochemical and structural properties of a protein, prediction of operands, gene ontology, evolutionary relationships, and metabolic cycles. Consequently, manual curation is a fundamental part of the process of assembling and annotating a genome, in which the curator is responsible for validating all of the predicted genes. A more detailed description of the gene or gene family product is obtained through similarity analysis using protein data banks that contain well-characterized and conserved proteins. Annotation generates data that allows various types of research on model organisms. After sequencing the organism's genome, sequences must be mapped according to areas pertinent to the research objectives. Gene predictions can be made with computational techniques for recognizing gene sequences, including stop codons and the initial portions of nucleotide sequences. This is known as functional annotation and can be done initially by computer using similarity in sequence alignment. However, no software is capable of generating a functional annotation without false positive results. Predicted genes need to be revised manually. When annotation is complete, the genome should subsequently be submitted to a public access site. Dear students, let's now talk about the data analysis pathways and tools. Alignment of sequences, bioinformatics, and molecular evolutionary analysis most often start with comparing DNA or amino acid sequences by aligning them. Pairwise alignment measures the similarities between a query sequence and each of those in a database using blast search. The most used bioinformatics tools, multiple sequence alignment is a useful tool in design experiments for testing and modifying the function of specific proteins, predicting their functions and structures, and identifying new members of protein families. Multiple sequence alignment of DNA, RNA, and protein is one of the most common and important tasks in bioinformatics. To process data efficiently, new software packages and algorithms are continuously being developed to improve protein identification, characterization, and quantification in terms of high throughput and statistical accuracy. In particular, for the analysis of plant proteins, extensive data elaboration is necessary due to the lack of structural information in the proteomic and genomic public databases. The high dimensionality of data generated from these studies will require the development of improved bioinformatics tools and data mining approaches. Dear students, let's now discuss about how to perform sequence analysis using software programs. Once we obtain a new sequence, or if we have a sequence of interest, we will see how we proceed. The stepwise sequence analysis can be performed using tools that are available on the web. Step first, to obtain sequence of interest. If you already have a sequence of interest, proceed to step second. In order to obtain a sequence of interest, we have to search PubMed or GenBank or SwissProt for obtaining the sequence of interest. Besides, we can also search a variety of proteins, nucleic acid, sequence, or structured databases till we obtain a sequence of interest. Once we obtain the sequence of interest, save the sequence in a separate file. Step second, identify open reading frames ORFs and translate into protein. If you already have a DNA sequence of interest, you need to be able to translate it so that you can perform further analysis. GeneFinder and GenScan, besides many more tools, are available to predict promoter, splice sites, translation initiation and termination sites. Moreover, XPASI and EBI are the simple translation tools available. Step 3. Find similar sequences in the database. Search the database of your choice using the BLAST search program. There are 5 types of BLAST programs available depending upon the nucleotide or amino acid sequence. Moreover, FASTA program is also available for searching. The FASTA search is preferred for nucleotide sequence and BLAST for protein sequence. Step 4. Do an alignment of sequences. The BLAST search engine provides local alignments, that is, the alignments of similar region. A global alignment, that is, the alignment of all regions, may help getting a better insight about your target sequence. Programs like Bayes, Block Aligner and Likelihood Weighted Sequence Alignment are used for the alignment of sequences. Step 5. Look for multiple sequence alignment. Clustral program is used for performing multiple sequence alignment with phylogenetic tree yielding capabilities. Saga, genetic algorithm, blocks, Macau are other tools for multiple sequence alignment. Step 6. Look for the presence of specific patterns in your protein. Search the ProSite library for profiles using the program Profile Scan Server at Strack. Blocks, Profile Analysis, Hammer, ProSite and PFAM are other programs to identify specific patterns in your protein. Step 7. Find similar sequences in other species. FASTA, SGD, that is the Saccharomyces cerevisiae database, ATDB, that is the Arabidopsis thaliana database, and several species-specific databases with BLAST can be searched to identify similar sequences in other species. Step 8. Determine the putative structure of your protein. PDB BLAST. In order to predict the secondary structure of protein, programs like JPRED, NNPRED, Predator and SSP programs can be used. For fold recognition software, programs like JPRED, 3-dimensional PSSM, Thruder2 and Toppets can be implemented. For predicting the structural features, programs like Modeler, Swiss Model can be used. Step 9. Obtain function of query proteins. To understand the function of your protein, it may be useful to gather information about proteins that share similar structural or sequence elements. By using PubMed search using as keywords the names of the proteins revealed by the various steps of this analysis may provide you with functional insight about your protein. And finally, input your sequence into an alert server. Alert servers will send you a message if a sequence similar to yours has been entered in any database. Sequence alerting and MIPS alert are the programs available for getting alerts. Dear students, finally we talk about the application of sequence analysis. First, the comparison of nucleotide and amino acid sequences is used to find the similarity and to understand whether they are homologous or not. Second, identification of sequence differences and variations such as point mutations and single nucleotide polymorphism in order to get the genetic marker. Third, identification of molecular structure from sequence alone. Fourth, to trace out the ancestral relationships and genetic diversity of organisms. Fifth, it helps in understanding the enzyme mechanisms. Sixth, it is very useful in clustering of families, superfamilies, and so on. Seventh, it helps in the identification of molecular surfaces in protein-protein and protein-DNA interactions. Eighth, it helps in metabolic computing, comparative genome analysis. And finally, it provides a direction for functional genomics, protein engineering, etc. This is the end of today's program. Hope you have enjoyed and understood it well. Thank you.